Naama received her PhD in 2014 from Weizmann, where she worked presumably on health effect of bioaerosols. She is now a postdoc in Max Planck Institute for Chemistry, and she'll talk about microbiology in the Earth system, the impact on environmental processes. To remind you, we also had a seminar at 12 today, given by Guy Dagan from Weizmann, and he'll be talking about clouds, aerosols, and climate.
Thank you for this, and thank you for the invitation to come here today. My talk will be today about microbiology in the Earth system, the impact on environmental processes. And I will go through a brief introduction into the field, and go then into environmental factors that impact microorganisms, such as CO2 in the atmosphere and urban air pollution. Move on to possible human health effects of microorganisms, like cyanobacterial endotoxins and allergens. And move on to the microorganism in extreme environments, such as in the atmosphere and in desert varnish in desert environments, and conclude at the end. So let's start with a brief introduction on a size scale on the microbiology world. We all begin with an atom at the range of 10 to the minus 10 meter. Bigger molecules than that are volatiles like isoprene. More complex molecules like proteins and lipids will be in the scales of nanometers. Then we'll see viruses, very small units, microorganisms, in the range of, to scale it to fine aerosols, a few nanometers, 10 to the 100 nanometer size. Bigger than that are bacteria and fungal spores that we find in the range, of course, also particles. Bigger bacteria will be the cyanobacteria, which would be the 10 micrometer sizes, in the range of small as crystals. And the bigger particles that we see, not considered as microorganisms, but still we look at them in an atmospheric environment, for example, are pollens emitted from trees and grass in the range of hundreds of micrometers. And role of microorganisms in the environment are diverse. They are abundant in every environment, actually. And mostly we see their impact in environments when we have limited photosynthesis because we have reduced light. For example, in the mid-Pacific Ocean, when we have black smokers emitting gases, we can find these tube worms, and they survive there and get their energy from small bacteria that can transfer sulfuric gases and hydrogen into energy. And that allows these tube worms to grow there. Other microorganisms like fungi will be decomposers. They will decompose organic material, dead plants and animals, and they produce nutrients and emit CO2 into the atmosphere, introducing that into the biochemical cycle of the CO2. Microorganisms are involved in different biochemical cycles, like the nitrogen cycle. For example, we have bacteria that can fix nitrogen to make it more available for more higher plants. For example, we find them in the rhizosphere, where we have the roots of the plant. And using new biological techniques, we use them for studying global biochemical cycles across many Earth compartments. Detection of microorganisms, let me sprint through that. Their traditional method originally started with Louis Pasteur, for example, looking through the microscope, and then we identify the structure, the morphology of the microorganism we want to identify. For example, here you can see Sphagius fungatus conidiophore, a very typical form of this fungi that you can identify through these figures. We can also sample them and try to regrow them and cultivate microorganisms in media. These are petri dishes. For example, here we sampled air sample in spring and try to cultivate what we see there in this filter and isolate the different species. But we know from genomic analysis then this is about 10% of what we actually have on these filters. So these are quite limited methods to identify and see what we have in our sample. And therefore, there are new advances in genomic approaches and that enable reducing the gap in identifying environmental microorganisms. And in my studies, I applied a lot of genomic analysis and tried to use them to learn more about microorganisms. So let's see, genomic-based methods, they are rapidly growing in recent years in use. They are based on an identification of specific gene sequences. Actually, we take the organism and look at the DNA. And then we don't care if the organism is dead or alive, if it's degraded, the DNA will be there and we'll see it. So we can detect the organism that used to live there in this sample using it, looking at the DNA. It's a very rapid and high resolution technique for identifying microorganisms. And as you can see here in the graph, sequencing or identifying the genomes of microorganisms started in the 1990s, where we have the first bacterial sequence in 1995, for example, and since then it has been increasing in use of identifying genomes, especially when we started using a very sophisticated technique of high throughput sequencing. We have really an increase, like a jump in usage of this method in studies. So let's see environmental factors that impact microorganisms and focus on the atmospheric CO2. We know that CO2 is increasing recently in the atmosphere, and a lot of studies have been conducted on seeing how that affects microorganisms. We know it affects correlation of fungi and plants pollination. We look also at earlier pollination timing and the amount of pollens emitted from plants. And this is important, for example, for allergic people who suffer from allergy to these pollens and start to suffer from that really earlier in the year. We know also that CO2 increase also impacts bacterial biomass, the reproduction and structure of the community. For example, in plants we see that really affects the microbial community or the microbiome around the plant. It affects plant yield and quality. So this is something important. And specifically in our study, we wanted to see how increasing CO2 levels might also affect allergenicity of fungal spores. We grew them in different CO2 levels. It was then at the level when the current CO2 levels were 394, now it's more than 400. We looked at compared to pre-industrial levels of 280 and doubling CO2 levels of 560. And we looked at different levels. For example, here you can see the level of the genes, when we have different genes encoding for allergenic protein formation. Most of them have a really high peak here, mostly the main allergenic gene here, the in red. And this is the corresponding behavior as the concentration of this allergenic protein that we extracted and detected, and general allergenicity, which we detected in immunological methods.
So we should double our CO2.
Yes, that would be. We think that it's really an interesting phenomenon that is really now in the optimum area. It may be that increasing CO2 also reduces the pH, and that's also affect the reproduction and productivity of these fungi. Also could affect respiration. This is something that needs to be further checked, but it's really interesting phenomenon. Air pollution is also a big phenomena impacting our health directly. We see that every day, but it also affects microorganisms, including their activity, growth rates, and this is something that has been done I reported previously. And in this study, we want to see if there is also an indirect effect of urban air pollution affecting microorganisms that affect our health too. And again, we looked at fungal spores that are allergenic, and we looked at their aerial lifetime when they are actually, after they are emitted, exposed to air pollution, and what's happened there. And we see that there are two things that happen. Both there is a changes, chemical changes in the proteins of these spores, and also the activity, the allergenic activity of this fungi is changed with time of exposure. And after 6 hours of exposure, it really increases compared to the control. We see more allergenic activity, and in this region of 6 and 12 hours of exposure, we see also nitration dominant changes of the proteins. Later on, we really see a reduction in activity in 24-hour exposure, 48, and it also corresponds to the amidation, which means actually starting of degradation of the proteins. We also know that microorganisms directly affect human health. In the case of cyanobacteria, they are really an important microorganism, have a lot of benefits, supposedly the ones who are in charge of the increase of oxygen level in ancient times. They're able to do photosynthesis, etc. But they also emit toxins, and this is a very known phenomenon from cyanobacteria. We call them exotoxin, and as they are excreted out from the cells, there are chemicals that induce different health problems. We find them, I don't know if you can see it in this figure, but there are some blooms of, for example, Microcystis in the Kinneret lake. And these are, the concern there is that this cyanobacteria will start also producing toxins, and that could affect both the animals living there, the fishes, and the other smaller animals, and also our health. Different phenomena include damage to the liver and pancreas, digestive problems, endocrinic problems, damage to the skin, and also to the nervous system. It depends on the different toxins from different cyanobacteria, but this is what we looked at was a different health effect by cyanobacteria. In general, cyanobacteria are considered as gram-negative bacteria, and that makes her composed, the outer shell composed of endotoxins. Part of the outer shell is endotoxin, which can also cause different health problems. For example, when we are sick, we have inflammation. This would be the cause for increasing fever and reactive oxygen species formation in our body, etc. So in here, what we asked was, is there a linkage between a cyanobacterial bloom in the Mediterranean Sea and endotoxin we detect on filters on the shore? And you can see really on the shore, we sampled the Untitled on the shore, there is a correlation between the endotoxin levels we detected on the filter and chlorophyll that was evaluated in the sea, and also you can see the trajectory is going through this bloom. We know this is cyanobacterial bloom, a very typical one in the Mediterranean. We also compared that to levels of cyanobacteria we detected genomically on these filters, and the high correlation is observed there too. But the interesting thing is that it really quickly, relatively quickly, degrades or changes when we see no correlation with endotoxin when we go farther inland to an urban environment in Rehovot. And we think that maybe this is due to either degradation, fast degradation of endotoxin when they transport dried in the air, and also impaction on walls around in surrounding environment, degradation through air pollution, etc. And this is something we are interested in looking forward. Another human health effect of microorganisms is allergy. Allergy is a severe phenomenon which we see an increase in trend along the years. Reported cases for, this is food allergy, but there are other cases for also, we call them aeroallergens, allergens that we breathe. Allergens include animal allergens. Allergens originate from plants, grass pollen, tree pollens, from mold or fungal spores, from insects, etc. And and we know that there are a lot of allergens that we don't know yet, they are not identified. And in this study, what we want to see is to look if cyanobacteria could also be one of these unknown or not yet well-defined allergens. And we started screening for different cyanobacteria originated from different environments, for example, from terrestrial environment, marine environment, and freshwater cyanobacteria, like the ones from the Kinneret. Grew them in the lab and really try to see immunologically if there is any active allergic effect from cyanobacteria. And while we did that, there was another publication reported phycocyanin, which is another compound in cyanobacteria. It's a part of the photosynthesis compartment in cyanobacteria, which also they reported to be an allergen. And the problem with this compound is that it's served in the industry as a colorant dye, natural colorant dye in food, for example, and any other usage, but when it's an allergenic candidate, it needs to be known and defined when we use it in food as colorant dye. So we try to both characterize allergenicity of this species and compare to see if this is also the main allergenic candidate in this species. And what you can see here, I show here inhibition assays, it's actually measure how similar our protein to the phycocyanin standard that we use. And you can see here for a terrestrial species that had also low allergenic activity observed and low similarity with phycocyanin. Marine species observed did show allergenic activity, but it was different than the one we saw from phycocyanin. And for the freshwater cyanobacteria bacteria, we saw both an active allergenicity and also similarity with phycocyanin. What we think is, in all cases, we saw in mass spec that we have phycocyanin in these studies, in these samples, and we should have because cyanobacteria have them, but the differences in the structure of phycocyanin is changed, and there are some studies on that between species originated from different environments. And this is what we think could be the reason for the differences between marine and freshwater cyanobacteria. So when we conclude that, we can say that terrestrial species, we can cluster them, that terrestrial species are a weak allergenic candidate, marine species are allergenic candidate with phycocyanin dissimilarity, and freshwater cyanobacteria are the most similar to phycocyanin in behavior, allergenic behavior. And I would like to move to look at the bacteria in other extreme environment, and these are a very interesting phenomena because this microorganism needs to be very creative in order to survive different conditions, like here in a hypersaline environment, like in the Dead Sea, very cold environment, like in the Arctic, and also we find them in hot springs, like in Yellowstone. And they have very unique characteristics that we can also take advantage of and use for example, enzymes that can be activated at very high temperature, like 95 degrees. We can then use them in application in our lab, and this is something was identified in such extreme environments. The atmosphere is also considered an extreme environment, Their airborne microorganisms are defined as bioaerosols. They experience very fast temperature changes, humid and dry conditions, wind shear, interaction with other reactive gases, other particles. It's a hard condition for them to survive in. And it's very interesting because it serves as a vector to transport these microorganisms from one environment to the other, from one environment to the other, so this is why we are interested in this medium. And we use different techniques to characterize different types of organisms in this in the atmosphere. For example, here what we did was to try to see seasonality and quantify fungal allergen, different types of fungal allergens using genomic analysis. We quantify the genes that we detected on these filters and try to evaluate seasonal distribution. So you can see here the relative abundance of specific species like Platosporium, for example, Donaria, etc., which are all allergenic allergenic spores in different seasons, winter, spring, fall, and summer. Quantitatively, we saw the highest concentration in the fall and in the winter, as it makes sense, it was expected. But seasonality, the abundance is not significantly so different. Really, you can see some difference, but the significance is not in the change in the composition of these allergens. We moved on. Another application, what we try to do here, we try to use the same air filter that we used and perform the metagenomic next generation sequencing, which gives us a bigger output of pathogens that we can detect. It gives us only the relative abundance of these pathogens, not the quantitative information. And what we tried to see here was if we can use combination of relative abundance compared with the quantitative input that we have from the quantification that we did before, and to give them the absolute concentration of this species in different seasons. And this is another study that we are currently wrapping up, also looking at plant pathogens or mycota, and try to see if we can link the identified species with different environmental conditions. For example, here you can see different temperature, and we cluster different species based on different temperature levels. And to conclude that, combining genomic finding with environmental factors like the temperature, seasonality, and others can help predict appearance of pathogens and allergens from an alien source. Another extreme environment is the desert. It requires adaptation to high temperature, dry conditions, radiation. There we find organisms in very unique surroundings, very interesting. For example, here you can see cryptogamic covers. It's actually a community of different microorganisms living together, surviving together in very hard conditions. It includes mosses, cyanobacteria, lichen, algae, etc. We also find them inside the rock. Here, for example, you can see a green area where the rock was broken and wetted, and suddenly cyanobacteria there started to proliferate and produce chlorophyll and do photosynthesis. And this is, we call them endolymphic microorganisms. And there we also find organisms in very unexpected environments, like in varnish rocks. And I want to zoom into this phenomena. Varnish rocks are mostly familiar to you, I suppose. You can find them in the Cacao Mountain, for example, in the desert. They are rich with manganese oxides, ferrous oxides in the outer shell, outer cover. As you can see here, this is the layer of the varnish, and this is the host rock. And it's a very interesting phenomenon. There is a debate on how it was formed. It's also interesting culturally. We see these petroglyphs there. We can learn more about ancient cultures there. We find it here. We find it in Saudi Arabia. And this is something interesting. And it's also being hypothesized to be detected in Mars. These are figures from Mars. Looking at manganese-rich rocks there, of course, we have to still validate that, bring them here in the study, but it seems that maybe there is a similar mechanism going on there, and if we understand what's going on here, how varnish is formed, maybe it can also give us some input on the condition it has to be in Mars to form there. Recently, there was a published paper on the types of varnish rocks we find. They can be found in different environments, like in the desert, in semi-arid environments, on building even, in river areas. And in our study, we only looked at the type 1 varnish, which is originated from desert area, structured, laminar structure, very fine-grained characteristics. And the debate about the formation has two main approach. The physicochemical process approach says actually dust is deposited on the rock and rain or humid condition bring to changes in pH, leading to separation of manganese from ferrous oxides, leading to oxidation of the manganese, clustering of manganese oxides, later on moving on and bringing that to form this varnish layer. The other extreme other side is claimed from a biology point of view that the varnish is actually formed due to microorganisms. We have microorganisms, we know that different bacillus species, for example, in the ocean are able to produce manganese oxides, and the claim is that this type of species are also presented in the varnish, actively oxidizing manganese oxides secreted out from the cells, forming the layer, and this is this explanation. This has not been proven so far. Microorganisms have been detected microscopically and assumed to be the ones who are in charge of varnish formation, but still finding them there doesn't mean necessarily that really they are the ones who form it actively. And in our study, what we want to see is that if we can give another input by looking at the genomic analysis of desert varnish microorganisms. So what we did was to collect varnish from different environments, mostly in California, but also in Israel and in Saudi Arabia. And we extracted DNA from varnish outer surface using a polishing device in certain condition, send it for identification. And I would like to say a few words on the technique we use here, which is a next generation sequencing. We take our DNA sample and try to see what we have there, to identify the whole DNA we have in the sample. It's currently in the front of the genomic techniques, produces very big database of DNA sequences. In our case, for example, we could have 35 million sequences per sample, which is a huge information. And then we need to know what we do with these sequences. We have a list of identified DNA sequences, and in order to sort it and make sense of this information, we need to use bioinformatics, and this is a very powerful tool to try and digest and get an input from this genomic analysis. And let me show you here some of the things that we have done with our samples. Use the varnish samples, compare them to soil samples that we extracted from the same area. And you can see here 3 soil samples collected from underneath the varnish rocks, very similar in composition. This is the composition differences. Compared to the varnish, 3 varnish samples extracted DNA, which is, 2 of them are similar and 1 is a bit different, but still very different from the soil samples, so you can really get an information on the differences in community composition. This is a very big graph with a lot of information. Let me go with you through that, but in general, we can take our DNA sequences, get the information, compare that to libraries of the organism, and get the information of the family and the phyla we see in our samples, get a phylogenetic tree of our sample. And what we did here was to compare the families that we find in desert varnish, compare them to the soil. And for example, in the soil samples, we see more DNA amount and more species. But as you can see, the round square represents the actinobacteria. There are more limited species observed in the soil sample, as in the varnish we can see cyanobacteria in gray, proteobacteria in much more abundance in the varnish compared to the soil. Another information we can get from this DNA is not only looking at who is in this sample, but also what it can do. The activity, the functionality of of the species, where we look at genus, specific regions in the DNA that will encode for protein formation, like enzymes, that maybe would be able to oxidize manganese. So we can see the potential of the species to oxidize manganese and see if there is any evidence for this species to be there. And what you can see here is a volcano plot, we call it. We have each dot represent different gene. If it's more abundant in the soil sample, it will be here in this region. If it's more abundant in the varnish, it will be in this region. We tested the differences in two different statistical assays. If it's significant in one assay, it's determined in dark blue. If it's significant in the second assay, it will be in light blue here. And if it's significant in both assays, it will be marked in green. And these are the genes that we are looking at, significantly different in both assays. And here, what we did was kind of a list of candidate genes that we have. Each line here represents a different gene. The colors represent the frequency of appearance. For the different samples, for varnish samples and for the soil samples, and we have a list, it's numbered, and I don't give you the list here because it's really long, but we screened through it very thoroughly and we could not find oxidizing genus that will be able to oxidize any transitional metals. So not only manganese, we could also not find iron oxidizers, etc. It doesn't mean that they are not there. It just means that what we currently know of from the literature, it's not here. And this is an interesting finding. We also tried to validate that in quantitative analysis with qPCR to see specifically different genes. If we find them, we didn't see any significant change. And then we concluded that we cannot see a relative or quantitative enrichment in manganese oxidizing enzymes. But we think, as biologists, that they surely have some kind of an input, these microorganisms there, to the formation of the varnish, and it might be indirect. It might be through, for example, cyanobacteria that we find there contains manganese in their cells, and when it's degraded, it might contribute manganese to the formation of the varnish. It also may change by respiration the pH that might affect somehow the varnish formation, but that is something that really needs to be further checked. Currently, we cannot say that there is an active formation of varnish through microorganisms. So let me conclude and say that I showed you that levels of atmospheric CO2 and exposure time to air pollution impact microorganism health effects. We saw that marine-originated cyanobacteria are associated with the toxin detected onshore. We saw that allergenic potential of cyanobacteria and similarity to phycocyanin can be clustered based on the growth origin of the cyanobacteria. And when we combine genomic findings with environmental factors, we can predict more efficiently appearance of pathogens and allergens from an alien source. And lastly, a high diversity of desert varnish microbial community was observed with no indication for direct microbial involvement in formation. And the take-home message for you is that biological tools are an advantage additive in earth science studies. Thank you.
It's interesting that even Morgan, in his many years old about how you mobilize manganese in an environment, he talked about two catalysts. One is the clay and the other some organic thing. The problem that the quantities of manganese in the rock varnish, if it was catalyzed by organics, we should have had an order of magnitude more. So already the early calculations show that the chance that these guys are doing it.
Yes, but yet a lot of other studies did show it is possible, and different organisms have different rate of formation. So it had to be proven, or it still has to be proven if it's there. And I have a lot of ideas of how to do that, but it's an ongoing study that there's a lot of things to do yet.
With regard to the effect of the CO2 levels, wouldn't they just evolve, or they are evolving all the time? So what you measure today is you see the adaptation for today's conditions, and if the CO2 will grow, the speed can
We
move.
also thought that this is maybe the case. We tried also to do that, but this is something that you need to generate for generations and generations of growth to adapt them for new CO2 levels. We tried to do that, but the time scale that I've been there, it was too short. But it's really also something, yes, that we thought of.
You seem to have found the whole zoo of bacteria on these dry desert pebbles. I just wonder, where are they? On the surface or embedded within the varnish?
We are not sure. We think most of the DNA will be in the upper layer of the varnish because it will degrade eventually with time. We have to see that, and the idea now is maybe to use some fluorescent detection to really have cuts of the DNA of the varnished rock, and try to label fluorescentically the DNA and enhance that, and try to see where we can locate the DNA there. This is the idea. But we think it may be in the upper.
And more of a comment. I think that when you talk about, for instance, you mentioned the atmosphere as being an extreme environment, and you missed, I think, one component which is crucial. This is sunlight.
I said radiation, but it's...
Yeah, but radiation is like almost under the rug. But you mentioned, by the way, radiation when you talked about the varnish, but I'm talking about the aerosols in general. I mean, chemical reactions releasing reactive oxygen species, abiotically, nothing to do with biological activities, also affect and also can be affected by activities of the organism which try to protect themselves from the damage.
Yes, I wrote that one of the reactions would be with gases in the atmosphere, but of course, yes, photooxidation and another reaction like that.
It also determines, it changes pH.
Right, in cloud environment, in dry conditions.